Anushma,
Yes,
start.
ma'am.
You can start, Bachcha. Start the session.
Okay,
Madam,
ma'am.
let us wait for another 2 minutes. Let the delegate cross 30, please.
Okay.
It is just 2.
If sir has
Sorry
no
to
problem.
interrupt you, Dr. Arun. Otherwise, you are very prompt.
No issue, sir. No issue.
ट्राई करती रहेंगी वो. हो गया ना. हाँ, वही उन्होंने बोला कि वेट कर लो अभी 2-3 मिनट और. हो गया. चलो, गुड. कर दिया मैम को.
Good afternoon, sir.
Good afternoon.
Madam has joined.
Hi, Geeta. Good afternoon.
Good afternoon, sir. Good afternoon. Sorry, I'm late. Actually, there is some internet issue. That's why I was not able to join on time.
Yeah, we were waiting for the same.
Yeah, thank you so much. Yeah. Manju ma'am, we can start the session, I think.
Yes, madam.
Yes. Yes, ma'am. Anushma?
Yes, ma'am. Shall I start the session?
Yes.
Okay. Good afternoon to all the delegates. Now I welcome you to the afternoon session of 35th QIP Day 3. I would like to welcome Dr. Geeta Agrawal, who will be chairing our session. Dr. Geeta Agrawal is working as Dean Academic and Professor of Pharmaceutics in Delhi Pharmaceutical Sciences and Research University. She is interim CEO for DIPSAR Innovation and Incubation Foundation. Dr. Agrawal has more than 16 years of rich experience in academics, administrative functions, and research. Her research area of interest includes targeted drug delivery with nanocarriers, transdermal and topical drug delivery, exploitation of modified natural polymers and lipids for enhancement of bioavailability of poorly soluble drugs, validation of packaging solutions, nutraceutical and herbal cosmeceuticals. She has keen interest in entrepreneurship development activity. She has completed 6 industry-sponsored projects and working on various research projects. She has successfully developed innovative and affordable validated cold chain shipping solution under a startup project funded by Incubation Foundation. She has filed 7 patents within 3 years. Her research products are commercialized, which includes hyperprotein, nutraceuticals, and validated shipping solutions. Dr. Agrawal has more than 120 publications in journals of repute in her credit. She has authored 5 books and 10 book chapters and has supervised more than 40 M.Pharm students and 3 PhD students for their research projects. Dr. Geeta has worked as scientific convener of national conferences and as organizing secretary of FDPS. She has worked as convener of academic planning and monitoring committee, research coordinator, academic coordinator, coordinator for PhD program, and finishing school. She is also involved in university industry lessons. Now, I would like to request Dr. Geeta Agrawal to kindly introduce our resource person.
Thank you for your kind words. Good afternoon, everyone. I am really pleased to introduce our today's speaker, Professor Arun Nanda. Professor Arun Nanda is very senior professor in pharmaceutics and presently working as professor of pharmaceutics in MD University, Rohtak. He is also director Aryabhatta Central Instrumentation Laboratory, MD University, and Professor Nanda has been associated with various universities as member of Board of Studies Selection Committee, Research Committee, UGC, AICT, PCI, CSIR, NBA, NET, etc. Professor Nanda is graduate and postgraduate from Delhi University and is alumnus of DIPSAR. So this is really good for us. He has done his doctorate from Jamia Hamdard, New Delhi, and LLB from MD University, Rohtak. Sir has guided various M.Pharm and PhD students and has more than 30 years of teaching and research experience. Professor Nanda has written various books, book chapters, authored more than 140 papers in journals of repute, and delivered various lectures in various topics in pharmacy. Sir, we are pleased to welcome you on behalf of university. We are very happy here that you are with us. And today, sir is going to speak on a very relevant topic, traditional knowledge and patent issues along with case studies. Again, welcome you, sir, and now over to you, please.
Thank you, ma'am.
Yeah, one thing I want to say, last month he received Excellence in Research Award from Indian Association for Reliability and Statistics. He received an award.
Great,
Excellence
sir.
in Research Award. Congratulations, sir.
Thank you, ma'am. Thanks for the
Now
kind
over
words.
to you.
Congratulations, sir.
Shall I start, sir?
Yes,
Yes.
yes, yes.
Yes, sir, please.
Good afternoon, all of you. In fact, I'm slightly out of context because I'm not a person from pharmacognosy. Still, I would like to share some of my views because one of my areas of interest has been the patents and copyrights and IPRs. They are applicable everywhere. Ma'am, is my slide visible?
Yes, yes, yes. It's visible.
Okay. So I'll be sharing some of my views on traditional knowledge and patent issues. In my presentation, I'll be briefly discussing the traditional knowledge. I'll be discussing the patent cases of turmeric and neem, and then I'll discuss how the government of India fought with this biopiracy or the patent cases, and ultimately something good has emerged out of it. That is traditional knowledge digital library. So to begin with, okay, we understand traditional knowledge basically is the knowledge which we have possessed over centuries, which is our traditional knowledge passed on from generation to generation. Could be in the form of stories, folklore, orally transmitted, community traditions, local languages, and so on. And in most of the cases, this traditional language, this traditional knowledge is not documented. Even when it is documented, it might be in some language like Sanskrit or Tamil, and that too in a dialect, which is an old dialect. So, India, as we know, is rich in biodiversity, and it is very unfortunate. When I focused on the patent cases, our friends, basically the Indians who are working in some places like US or some universities abroad, they are the persons who are sharing the biodiversity of India, getting it patented, and thereby denying us the benefit. So I'll start with the case study of turmeric. If you focus your attention on the inventors, Soman Kumar Das and Harihar Prasad Kohli, the spellings are different in US, but then basically they are Indians. They got a patent on turmeric in 1995, and the turmeric which they used for their patent was purchased by them from a departmental store. It was edible grade turmeric. Their basic claim was the beneficial use of turmeric on wound healing given by the oral route or used topically or a mixture of both. And upon grant of patent, the patentees got an exclusive right to sell and distribute turmeric. Had this patent continued, it would have been next to impossible for any Indian to buy turmeric from the local kirana shop or the departmental store. I repeat, they had got the patent rights on the use of turmeric from kirana shops. And the problem is that if I'm buying turmeric from a departmental store, that is domestic grade turmeric, even that would have been covered under this patent. And this is something which really shocked the government of India. I'll trace out the this patent in details. So basically, it had 6 claims. They had they claimed that patent this turmeric is a wound healing agent. It could be administered orally. It could be administered topically. It could be administered in a mixture, oral and topical. And the wound, they said here, was a surgically inflicted wound. And they also claimed that they were successful in a limited clinical trial. They used 2 patients, and they said they have been able to heal a body ulcer. So, in the turmeric patent, they had given some of the prior art, and the use of turmeric as a wound healing agent in India is well known. It is our traditional knowledge. We are using it over generations. But the problem was that when this patent was awarded at the US Patent Office, the US Patent Office has some typical requirements: novelty, utility, and innovation. The patent the applicants could satisfy all the 3 applications, and the US Patent Office awarded them the patent. At that time, since US is a party to TRIPS and India was also a signatory to TRIPS, that is Trade Related Intellectual Properties Agreement. Therefore, this patent was applicable on us. And the government of India was really shocked how this patent could be awarded. At that time, when the government of India tried to represent to the US Patent Office and tried to oppose this patent, there were many problems. Now, as on date, the procedure for opposition of patent in US is quite simple. But in those days, that is when this turmeric patent was awarded, Indian government was a third party in the US Patent Office. Although we represented our opposition, but we could not participate directly. It is very interesting that this patent was filed by 2 Indians. The patent was examined by an Indian. The patent was re-examined by an Indian, and the patent was opposed by an Indian agency. One more thing, when this patent was awarded, I was in Delhi. CSIR was looking for documented articles on use of turmeric on wound healing, and that too a surgically inflicted wound. So I remember in those days there was a they could get hold of one research paper in Persian language, that is in Indian Institute of Islamic Studies, wherein it was mentioned that one soldier in a war had a wound. They described it as nashtar ka ghav. Nashtar is a sort of a fighting sword. So nashtar ke ghav के ऊपर हल्दी छिड़की गई. The problem which we faced while opposing this patent was US Patent Office has a requirement that if you say use of haldi is documented in our traditional knowledge, they will say bring some printed information, bring some article. If you say no, our forefathers know it, we have been using it for generations, this is a part of our traditional knowledge which is transmitted orally, US Patent Office does not recognize this. So this was a typical patent case wherein the government of India and the agency CSIR had a very tough time in collecting documented articles on the use of turmeric in surgically inflicted wounds. And you may be surprised that they could get only 32 research articles on the use of turmeric in some journals. Ultimately, upon presenting this information, the US Patent Office was satisfied and this patent was revoked. But this patent really shocked the government of India, and because of this patent issue, because of this problem of biopiracy, infringement of the misuse of traditional knowledge by Western agencies, the government of India took two drastic steps. Number one, there was a modification, there was an amendment in our Indian Patents Act in pre-grant and post-grant opposition. I'll focus on that. And the second most important part was the development of a traditional knowledge digital library, wherein the government of India tried to get all the traditional knowledge documented. Because once you have anything in a document, and if this document is in public domain, that is, it is easily accessible to anybody, then this this would nullify one of the essential criteria of patent. Because if I make an invention and if I publish it, it is in public domain. Now, anything in public domain is not novel. It cannot be patented. So, with this patent, ultimately the government of India woke up and took steps how to prevent biopiracy, how to stop biopiracy, how to stop the misuse of traditional knowledge. Okay. So, the summary of this invention was that the inventor said we could use turmeric for topical application and for oral use to promote wound healing. What they did was they took some rats and using a circular instrument, they inflicted some wounds, some precise wounds on some muscular part of the rats. Then the rats were distributed into various groups. The wounds which were inflicted, they were photographed, the biochemical parameters were noted, and they divided the rats into certain groups. In one group, that was the control group, they applied only normal saline over the wounds. In the second group, they applied oral turmeric by the oral route. In the third group, they sprinkled turmeric over the wound. In the fourth group, they gave turmeric as well as turmeric by the oral route as well as by the topical route. At the end of the experiment, they documented that whenever the rat was receiving turmeric by the oral route or by the topical route or by both, wound healing was better. this was documented by the area of the wound and by the use of the biochemical parameters. Then they also made an experiment wherein they thought that if we are using turmeric over a wound, will turmeric allow the cells to multiply, or does it have an opposite effect? So they took some placental cells, and in a culture, they allowed the placental cells to proliferate in presence of turmeric. And what they found was turmeric actually helped the cells to multiply, that is, cell proliferation. So, in other words, they said turmeric has no side effect upon the growth of the cells. Uh, then they took hold of two patients who had some wounds which had developed into some kind of body ulcers. They administered turmeric to those two human beings orally and topically, and they claimed that in both the patients the body ulcers were healed. With this experimentation, they could get a patent in US. So this was their theory that turmeric could improve microcirculation, stimulate angiogenesis, and so on. Uh, this is how they uh, conducted their experiment. Turmeric was used in the form of a powder which was obtained from a food store. This was a Raja store, Raja Departmental Store, Illinois. And this was the uh, heart of this patent, because had this patent continued, it would simply be translated as use of turmeric from when I'm purchasing turmeric from any departmental store or any kirana store. It would be very difficult for me to bypass this patent and to explain that I'm not using this turmeric for wound healing. Okay, so this was their experiment. They used 32 rats, surgical wound by a circular some scalpel type, and they distributed the animals in different groups. One group control, second group topical turmeric, third group oral turmeric, fourth group oral plus topical turmeric. They said the mean wound size was better when turmeric was used. They also photographed the progress of the wound. They said the wound management is better when turmeric was used. Then this is the case history of two patients, case history 1 and case history 2. In one patient, they said turmeric was effective in 4 weeks. In second patient, it was a couple of months, but then they said the skin ulcer could be healed when this skin ulcer was not responsive to many other treatments for these two patients. Then this was their experiment for determining the effect of turmeric on the endothelial cells of human umbilical cord, and they made an inference that turmeric has a proliferative effect on the endothelial cells. So in other words, it will stimulate the cell growth. So this patent was awarded in 1995. In 1996, CSIR challenged this patent and asked for re-examination. And specifically, CSIR asked for complete cancellation of all the 6 claims. There were long arguments, and ultimately, in August 1997, the US Patent Office revoked all the claims and cancelled this patent. The basis of the CSIR's opposition was that in India, use of turmeric of turmeric as a wound healing has been well known over the centuries. But it was a typical requirement of the US Patent Office that they said no, traditional knowledge is not a sufficient ground for us. Use of turmeric as a wound healing agent should be documented, and ultimately CSIR could produce 32 research papers. While discussing with a leading journal Science, one of the patentees, Harihar Prasad Das, he said he knew the traditional use of turmeric and he was simply trying to test its efficacy. And he said, I have also used clinical trials. According to Das, he said, I wanted this compound to be popularized, and one way was to develop it by using a patient. The opposition procedure was quite tedious, and government of India had to employ a US patent attorney, and they had to spend something like 15,000 US dollars. And there was a long wait. At that time, the opposition procedure in US was quite strict. Indian government could not participate directly. We were a third party uh, opponent. It means that the government of India or the nominee, that is CSIR, they were sitting outside, and it was only the US government, patent office, and US attorneys and our uh, attorney who were participating in the opposition proceedings. The problem was that India as a sovereign state was one of those affected, and in fact, all member states of TRIPS were affected. I would repeat that had this patent continued, the use of turmeric in kitchen in India could have been prohibited. So this was really a patent which jolted, I would say, the government of India. Once there was an opposition and it was successful, the University of Mississippi Medical Center, who was an assignee to the patent, they decided to abandon the patent. The patent was given back to the inventors. But though these two inventors, Kohli and Das, they went a step further and they wanted to pursue their case. They said the powder and paste had different physical properties, and therefore any person with a normal degree of art, normal skill, would not be in a position to find out the application, and therefore they said this patent should be continued. Further, they said orally turmeric is given with honey, and honey itself has wound healing properties. But US Patent Office rejected this opposition, and they accepted the CSIR's citations, and ultimately this patent was the claims were rejected a second time, and in the second re-examination, the patent was finally cancelled. In this patent, the patentees were Indians. The patent examiner in US Patent Office was an Indian, and the opposition proceedings, they were initiated by an Indian institution. You could see that we had to spend a lot on this unnecessary patent opposition. So basically, this patent actually transformed many things. I'll be discussing that after this patent in 2002, some amendments were made in the Indian Patents Act in the section 25 relating to pre-grant opposition and post-grant opposition. So I'll be discussing those oppositions also. those amendments also. Broadly, I would say there are three leading cases which transformed how the government of India tried to defend the traditional knowledge and the biodiversity in India. One of them was turmeric, the other was neem, and the third was rice patent. I'm not discussing rice patent today because of paucity of time, but I'll be sharing some of the information on neem. So we all know neem tree. We know it as Sarva Rog Nivarani, curer of all elements. It has been mentioned in Indian texts over 2000 years ago. It's a part of our culture, religion. In 1928, first report on pesticide property of neem was reported. Farmers in India have been traditionally using neem as a pesticide, and typically what these Indian farmers would do, they would take the neem seeds, crush them, and mix them with water. And this sort of suspension, it is rich in azadirachtin. But one of the major problems of this seed extract, aqueous extract, is the instability. But the Indian farmers know this. So they would crush the neem seeds and suspend it in water and immediately spray it. So the main problem of neem or the azadirachtin preparation is the stability. We know that apart from the pesticide properties, neem tree is also used in several other indications. Even now, people are trying it for use as female contraceptive, spermicide, anti-malarial, and so on. In 1959, one German entomologist reported that locust swarms did not harm the neem tree. That is, there was something in the neem trees which repelled the insects also. So farmers in India know that if they use alcohol, then this seed extract is somewhat stable and the efficacy is also better. But the Indian farmers have been using the seed extract traditionally, and that too over centuries. In 1992, US Patent Office issued a patent to W.R. Grace and Company. This is known as Grace Patent. This was on a derivative of neem tree. Grace Patent had two parts. One was a stabilized azadirachtin in solution. They used some A-protect solvents, DMSO, DMF, and so on. And in the second part, they used alcohol. So in both the extracts, they were quite stable, and they said since these extracts are stable, therefore they are more valuable to the pesticide industry. And in fact, Environmental Protection Agency also registered demix as the first product derived from neem tree approved as a pesticide in United States. This is the front page of the US patent, and if you look carefully, again we have an Indian here, Narendra Prasad Luthra. So it is our Indian scientists only who know the traditional knowledge, who are aware of this traditional knowledge, and who are trying to get it patented. So this was storage stabilized, stable azadirachtin formulation. And there were two main things. One is 50% by volume of A-protect solvents, such as DMSO, DMF. And in the second embodiment, it was more than 50% alcohol, some water, and some surfactants. So by the use of surfactants, they could make a stable emulsion. So according to US, purification or modification of a naturally occurring compound can get you a patent. they could satisfy the requirements of a patent. Practical utility was there. There was some novelty. They said there was no prior art in which the other directed suspension or solution could be stabilized. And this was not obvious from the prior art to a person of ordinary skill in the art. And they said we have given a complete description wherein anybody can use our patent or our formulation very easily. The fact that this improvement was obvious in India to Indian farmers, this did not help us to defend the patent in US, because one of the major problems in patent office of US was they said no, if you say this is a part of our traditional knowledge, we don't accept it. Give us a documentary proof. Now in India, one of the major problems is we know the use. Where is the use? It is in our head. We have not documented. And the US patent of 1992, this could not be approved. In fact, at that time, one Indian scientist said the patent on neem extract is as good as getting a patent on fire. So if anybody lights a fire, you will have to pay the price. Anyhow, but then these Americans, they went a step further. They filed a European patent application with the European Patent Office, seeking a priority application from US, covering a method for controlling fungi on plants by using hydrophobic extracted neem oil. Hello, I believe
Madam, please
Yes, sir.
somebody has to mute his audio.
Yeah.
Okay. In the European Patent Office, the examination was very strict, but still they could get a patent for a method for controlling fungi on plants. In 1995, there was a legal opposition against the grant of this patent in European Patent Office by one of the members of the European Parliament, then Dr. Vandana Shiva from India, on behalf of Research Foundation for Science, Technology and Natural Resource Policy, New Delhi, and International Federation of Organic Agricultural Movements based in Germany. They submitted that the fungicidal effects of the hydrophobic extracts of neem seeds are well known. They have been used for centuries in India, both in Ayurvedic medicine, and it is well known in India. Since this traditional knowledge was in public use, therefore, the opposition procedure in US Patent Office and in European Patent Office is different. US Patent Office insists upon documentary evidence, whereas the requirement of European Patent Office is not so strict. European Patent Office has one more peculiar thing. In this patent, the patentees were focusing on Aza directa indica. The European Patent Office says that whenever by award of a patent, if one plant is getting patented, that would mean monopoly. And normally they don't allow a patent on a single plant. If it is I would say a complete genus, then they may allow, but not the use of a single plant. So in 1997, the Opposition Board of European Patent Office summarized that this patent cannot be maintained in view of the evidence, and they also said there is relevant prior art. Then this was again examined, and they said all the features of the present claim have been disclosed to the public prior to the patent application during field trials in 2 Indian districts in 1985 and 1986. And they also said that it appeared a mere routine work for a skilled person to add an emulsifier in the appropriate amount, and therefore there was no innovation. So after a 10 years long battle, India won uh, this patent, European Patent Office, and this patent was denied. The government of India successfully argued that neem is a part of traditional Indian knowledge over 2000 years, and European Patent Office agreed to the claim of the government of India. I said the situation is different in European Patent Office. Even if we are not having any journal articles, but we are able to prove that yes, neem is a part of our traditional knowledge, we could oppose the patent. So broadly, after these patents, it emerged that if India wanted to protect its biodiversity from the biopirates, then two things were required: amend the patent laws and codify all the traditional knowledge, so that we could challenge any attempt to patent indigenous knowledge. The only, for example, all of us know the beneficial effects of tulsi. Now, if these beneficial effects are documented, they are written somewhere, and this information is in public domain, then anywhere in the world we can get this, uh, get a patent on such tulsi. We can oppose this patent. But the problem in India is that traditional knowledge as such, if it is available in Sanskrit, Tamil, then a US Patent Office examiner may not have access to it, may not be able to understand it. And therefore, in the first step, they will issue a patent. Later on, we'll have to spend money, we'll have to wait, and we'll have to fight this patent. So, what the government did, the first was the 2002 amendments. This is pre-grant opposition. That is when the patent has been published, but the patent has not been granted. Then there could be pre-grant opposition. And one of the grounds which was added here was that the invention so far claimed was anticipated having regard to the knowledge, oral or otherwise, available within any local or indigenous community in India or elsewhere. So this was a, I would say, a drastic step, wherein the traditional knowledge was included as a ground of the pre-grant opposition. This ground was also continued in the post-grant opposition, that is, when the patent has been granted, but before the expiry of the period of 1 year. And in fact, the uh, patent cases in India very well exhibit that the post-grant opposition is not limited to 1 year. Even when the patent has been awarded 10 years back, but if it can be proved that this knowledge was a part of the traditional knowledge, the patent would be revoked. Then uh, the government of India formed a traditional knowledge data library expert group in 2005. They informed that around 2,000 patents have been granted around the world erroneously based on Indian system of medicine by patent officers internationally. So this was a pioneering initiative by the government of India. It was initiated, it was flagged by the US patent on turmeric, then also supported by the rice patent and the neem patent. And the time, effort, and money spent on getting the patent on turmeric revoked at US Patent Office. This highlighted the need for a proactive mechanism of protecting the traditional knowledge. So this traditional knowledge digital library, what it has done, all the knowledge which we have in Sanskrit, Hindi, Arabic, Urdu, Tamil, etc., this has been published and it has been classified. And this classification is also matching with the classification of the international patent classifications. Then this traditional access to this traditional knowledge digital library is provided in 5 international languages, and 13 international patent officers have access to the traditional knowledge digital library. We have an MOU with them. The patent examiners in these 13 international patent offices can have access to our knowledge, but they cannot publish this information on their own. And as on date, more than 3.6 lakh formulations and practices have been transcribed into the traditional knowledge digital library. So basically, this has helped many international patent offices, and the government of India could find out that approximately 230 patent applications have been set aside or withdrawn or amended based upon the evidence of prior art on the basis of traditional knowledge digital library, whereas we had to spend around 15,000 US dollars for the turmeric patent and around 7 crores as a legal fees only for getting the patent on basmati revoked. So this is a screenshot of the traditional knowledge digital library. This is not a live page, but if you log in into the traditional knowledge digital library, and if you click into 1, 2, 3, 4, 5, 6, then you will get something like this: abstract, title, bio, bibliography, and used in the treatment of this, and so, and you'll get all the keywords and details. So with this, since the information which is a part of our traditional knowledge has now been documented, therefore any such patent based on our traditional knowledge will not be novel, and the international patent offices can have access to this in their own languages, and therefore they can oppose the or they can reject the Indian patent. My request to all the participants here would be that these 3 patents have highlighted one thing in common, that is, we Indians know many things, but this knowledge is in our brain. We hardly document it. So my request to you, all of you, would be whatever experiments you are doing, document them. And if you think you have to go for their publication, go for their publication at the earliest. If you think you have to get them patented, then file a patent first and go for the publication later on. And the second request would be that we start using this traditional knowledge digital library as part of the routine literature survey for our research projects, because the number of hits, not only do we get useful information, but if we are getting number of hits more, then probably this library would be expanded more. So with this, I come to an end, and in fact, this is going to be a reality, that this day is not far when our normal flora or normal vegetables, everything would be patented if we are not able to guard our traditional knowledge. We are not able to protect our traditional knowledge. So with this, I would end my presentation, and I would invite questions from the participants. Thank you.
Thank you so much, sir. It was really very, very informative, and personally, I found it very interesting too. And if I say you have explained both the case studies in detail, and I'm sure this is going to be very much useful for our students, research scholars, as well as all the participants. So I will again ask the participants if anybody wants to ask any question. They can unmute themselves and can ask directly, or they can type in the chat box. Yeah, as sir has told, it's really important to understand every aspect of it, like what is biopiracy, what is the governing act to prevent the biological resources of our country. So everything is important, sir, as most of the students and faculty members presently they're working on the biological resources and various traditional medicine systems. So it again becomes very important. Sir, I would also like to ask, as you have already told that there are certain issues in patenting the traditional knowledge. So what are the ways actually how we can pursue for that, that at some level we can protect it and we can efficiently work over it?
The first thing is that anything and everything which you are doing, get it documented. And when I say get it documented, it means the best thing is that you are getting it published in a journal. But even if you are making it a part of your MPharm or PhD thesis or your project report, even that is documentation. And I
Right.
would say that if your uh, method of documenting your research in your day-to-day copy, if that is clear, even that can form a basis of the opposition to the grant of a patent. You see, when CSIR tried to look for articles on use of turmeric for wound healing, they could get only 32.
Right.
And some of them were in Persian, some of them were in Urdu, some of them were in Tamil, and they had to translate it. So, do you think that 32 articles on use of turmeric in wound healing, is it sufficient? It is not
Never.
matching. It is not matching with the utility of turmeric in our day-to-day life. So this was a major problem, that we have not been able to document our traditional knowledge. And in fact, let me tell you, uh, please, this is not an offense to pharmacognosy, but myself, personally, I am very weak in pharmacognosy. I could hardly grasp the knowledge of pharmacognosy. But when I started my research, then I found that pharmaceutics or chemistry, this is no barrier. So, for example, transdermal drug delivery systems was one of my favorite research areas. I was using ultrasound. Now I'm using ultrasound and microwave
Right.
for novel drug extraction from plants. So, in a way, I'm mixing the pharmacognosy and pharmaceutics. So, this is my request to everybody: open up your barriers, open up your fields, go into the room of your colleague from some other field and associate yourself with them. You will find your research to be more productive.
Right, sir. This is great
Okay.
message. Yeah. We need to promote actually interdisciplinary research for better outcome for our students as well as for our country too. So this is really important. I think some student has asked the antifungal formulation patent included as well in the US. Parth Sharma. This is I am not able to understand the question. It is written by Mr. Parth Sharma. The antifungal formulation patent included as well in the US.
Ma'am, this US patent, we could not get the US patent revoked, but now it has died its own death. That is, now this patent has expired. At that
Yeah.
time, we could not get the US patent revoked. get the US patent revoked because we could not give them a documentary proof of how to stabilize azadirachtin extracts.
Right.
Okay, so at that time we could not do it, but now this patent has lapsed into the public domain. Now it's not a problem, and now it has been documented also. So now, still, I would say that if you uh, put some keywords, extra patents on neem, patents on uh, rice. In India, you would come across several such patents. I could get around 36 patents on different aspects. So I would say that award of a patent is not a problem. Patent once awarded, पत्थर की लकीर नहीं है. Because who is examining the patent? A person like you or me, who is
Yes,
a
sir.
patent examiner. So there is always a chance of error. And therefore, if somebody says so and so patent has been granted to this person, I'm not impressed. If you wish to impress me, then tell me, has your patent been actually used? Has
Right.
your patent been actually commercially utilized? If your patent is commercially utilized, then I would say, yes, you have done a wonderful thing. I'm getting a patent, putting it in a frame. Let me also share with all the participants here that if you are having a patent and if you are not using it, technically the patent can be revoked because every year you have to submit a utility report of the use of patent to the controller of patents. But in India, normally the controller of patents is not taking any action on unutilized patents. Okay, so it is not important to simply get a patent. It is equally important to utilize this patent.
Right, sir. Right. Thank you.
Any other question?
Yeah, there is another question. What about neem seed oil? Is it covered by patent?
See, I'm not very clear about this part. I do have an article with me, and I think just give me 1 minute, and I'll give you a list of some patents on neem, and I'll share it. Patents on neem.
Hello. Uh, Hello. Can I
I'm
ask?
sharing it, ma'am.
Yeah.
Yes, sir. Sure.
Hello, Professor Nanda, sir.
Yeah,
I'm
yeah.
very thankful for your very informative lecture. I enjoyed your session. I just wanted to
Thank
ask,
you.
we have done some work on curcumin a few years back, and like say, right, starting from a review to research work. So I would like to know that you have said that there are only 32 papers in this curcumin, regarding curcumin. So apart from wound healing, what other research prospect that you can share with us that we can work upon in curcumin? Apart from wound healing activity, which are the further research prospects that can be done on this curcumin or curcuma longa?
Ma'am, I'm not quite knowledgeable about pharmacognosy, I said. But yes, I do understand that now curcumin is being tried. In fact, like I'm working on co-crystals. That is a method to improve solubility. And one of my students is now thinking of using curcumin as a drug, and will be trying to make a co-crystal of curcumin and improve the solubility so that the bioavailability or delivery of curcumin, pure curcumin can be improved. So it's not wound healing only. I'm not clear about this part. I just know that yes, I have a molecule curcumin. Bioavailability is poor, so I can think of ways as a pharmaceutics person how to improve this part. Okay.
I think, sir, I'm from pharmacology department and I go main for the pharmacological activity. So viability was the main difficult part in this thing. Like say earlier, the piperine was included alongside with it to improve the viability of curcumin. Piperine was the component which used to be adjunct with this curcumin to improve the viability. But afterward, like, we were having a major problem in the solubility itself and the viability. So this work is on the hold for a while, but I would like to try for some other activities apart from wound healing for curcumin.
No, ma'am, I'm not working in this area. My present research areas are methods of improving solubility and improvement of bioavailability. So I'm using lipid carriers. I'm working on co-crystals. I'm working on solid dispersions and so on.
Okay.
And
Then
here I'm
session.
hardly knowledgeable about uh, the applications part, because traditionally I would select a molecule which is well known. Like I produced around 4 PhD theses in pharmacognosy. What we did, we took a typical plant. For example, this Pterocarpus marsupium, Kinu plant. So this is well documented for anti-diabetic use, right? We have it is a hard wood. इस पेड़ का तना जो है वो बहुत हार्ड होता है, and in market you will get some cups of Kinu plant. So what people would do, they would fill up this cup with water, cover it overnight, and in the morning they'll drink it. So the pharmacopoeia says maceration is a typical method for preparing extract of this Pterocarpus marsupium. What we did, we took the hardwood, applied ultrasound, and at that time I did not have the ultrasound probe, so I used the ultrasound basket only. And we also used microwave. So we had a novel extract, then we prepared a normal extract using maceration. Since this is a well documented plant, the anti-diabetic properties are known, then we compared the biological activity of these two extracts. And what we found that the novel extract was better. The novel extract had more active constituent and more biological activity. So in a way, I was able to uh, use pharmacognosy and pharmaceutics together. Okay, so here I would not go for a molecule which is not well established. Typically, I go for a molecule which is well known but which has a problem, like 7 days maceration or some percolation, and I'll conduct the same using a novel method in half an hour, or I'll try to conserve energy by green chemistry. So that way, I'm trying to get my earning out of this.
Okay, so thank you so very much. It was a very informative session on curcumin. Thank you so much.
Thanks, ma'am. Have a nice time. Thank you.
Thank you, sir. We have also got many compliments from the participants. Very interesting lecture, very informative. So thanks again
जैसे
for
इनका
this
बायोडाटा कैसे हटा है?
informative session.
Okay, thank you, ma'am. Have a nice time.
To
Thanks.
you, ma'am, please.
Thank you, sir, for your informative session. Now, I would like to request Dr. Geeta Agrawal to kindly felicitate Nanda, sir, and ask Mrs. Manju S. ma'am to kindly share e-certificate.
This is for you. Thanks for joining us during this QIP.
Thank you, ma'am.
Thank you very much, Dr. Nanda, for giving such a good
जी, ma'am.
presentation.
Thank you. Thank you, ji.
Thank you, Ji. Thank you.
Incidentally, Madam Manju and myself, we are batchmates.
Oh, great. ये हमें नहीं पता था. Sir, I have already listened to your many lectures
Okay.
about patents and it's really good. Thank you.
Thank you. Ma'am.
Thank you, Dr. Nanda. Madam, we have with us our next speaker,
Yeah.
Dr. Rajeshri Patel. She has joined. Welcome, ma'am.
Now I would like to request Geeta ma'am to kindly introduce our second resource person. Ma'am, kindly introduce.
Thank you. I am
Just a minute.
the second speaker of this session, Dr. Rajeshri Patil. She is the director at GCP Compass Limited, UK. Dr. Patil has worked as medical officer in various polyclinics such as patient care and medical records maintenance. He has worked as general practitioner as well as in clinical research in India, Singapore, and UK. She has experience in working as fail-safe officer in diabetic eye screening program in National Health Services, UK. She has worked as clinical researcher coordinator with Quintiles, India and Singapore based at hospital site. I welcome you, Dr. Patil, and we are eager to listen to you on the topic drug discovery and development in pharmaceutical industry. Over to you, ma'am.
Thank you. Can you hear me?
Yeah.
Yes, ma'am.
I'm just sharing my screen with you.
Yeah, sure.
Can you see my screen?
Yes.
Yeah, it is just start. Yes, it's visible.
Hello, everyone. Hope you can hear me well.
Yes, ma'am.
Hi, good afternoon. First of all, please allow me to thank DIPSAR to have this opportunity and to connect with you all and share some information related to clinical research. I have a few slides and I'll use them to present my thoughts. Moving on, next slide. Now, we all have experienced and still experiencing the impact of COVID-19 crisis. And while there are global efforts to get the control over this and get personal lives and business moving, we need to be prepared for such crisis in the future. Now it's COVID-19. Tomorrow it may be something else. And that may mean another outbreak of similar nature or maybe another pandemic or even a natural disaster like earthquake, wildfire, storm, or maybe tsunami. The list may go endless. Although we can learn from our past experiences, the challenges such as calamities will bring are not always likely to be the same. And this is where I would like to provide my thoughts as to how one can modernize clinical research and clinical research education by ensuring global requirements and, more importantly, respecting regional or local considerations to deliver proactive quality. Now, technology has become a big component of today's learning. Take today's example, where we have many participants all across India and some presenters like me presenting from thousands of miles away using this technology. Yes, there are some limitations where we are not sitting actually next to each other. However, we are still able to get same experience through this virtual world and get learning by sitting just at home. Now moving on next slide. What we are going to discuss today. So some topics we are covering today. First of all, we will see what are the top causes of deaths worldwide. Then we will see what is the drug development process. Next, we will go how the clinical research is evolving. Then we will see how clinical research education is evolving. And when this clinical research is evolving, we will see what are the career opportunities in clinical research for pharmacy profession. And the last, we will have key takeaways. Now, moving on next slide. Let's have a quick overview on top 10 causes of death. So now before we learn about good clinical practice, which is commonly referred as GCP, it is important to understand what is the clinical research and what is a clinical trial. According to World Health Organization, WHO, 56.9 million deaths occurred in 2016 globally. 54% contributed by the top 10 causes. Now let's have a look. Ischemic heart disease and stroke are the world's biggest killers, almost 15.2 million deaths, the leading cause of death globally in last 15 years. Next is chronic obstructive pulmonary disease, which claimed 3 million lives. Now the next is lung cancer, which is along with trachea and bronchus cancers. It caused 1.7 million deaths. Now next is the silent killer diabetes, which killed almost 1.6 million people in 2016, which was actually 1 million in 2000. Now deaths due to dementias are more than doubled in between 2000 to 2016, making it fifth leading cause of global deaths in 2016 compared to it was 14th in 2000. You can see the difference. Now the lower respiratory infections are the most deadly communicable disease. 3 million deaths worldwide happened in 2016. The death rate from diarrheal diseases decreased by almost 1 million between 2000 and 2016, but still caused 1.4 million deaths in 2016. Next, the number of tuberculosis deaths decreased during some period, but still among the top 10 causes with a death toll of 1.3 million. Now, HIV/AIDS is no longer among the world's top 10 causes of death, having killed 1 million people in 2016 compared with 1.5 million in 2000. And the last one is the road injuries, which killed almost 1.4 million people in 2016. About 3/4, which is 74% of whom were men and boys, unfortunately. Moving on to next slide. Now let's take a quick overview of drug development process. Can you hear me?
Yes, you are audible.
Yeah, thank you. Yeah, there was a noise in between, so I thought I'll try. Yeah. Okay, so we will see the drug development process now. A new drug development
Dr. Patil, ma'am, you unmute yourself. Dr. Patil?
Yeah, can you hear me now?
Okay, ma'am.
Yeah, now you are audible. Yes, please.
Yeah, okay. So let's move on. So we were talking about the drug development process, and a new drug development is a scientific effort which is highly regulated because of legitimate public health concern? I want you to remember that. It is a scientific process and it is highly regulated process because of the need to protect the general population. If you were public health officer or FDA monitor, you would be very concerned about releasing a drug that may not be efficacious or may not be safe. Very important to respect legitimate public health concerns. So the concepts of clinical research have their roots in highest level of ethical standards and patient safety. By this way, regulatory authorities are assured that all data supported to approve the drug is high quality and credible. I'm not going through all the lot of details of pharmacokinetics and pharmacodynamics, as I'm sure being a pharmacy professionals, you are very similar with all these concepts. Now, moving on next slide. Now let us have a look how the clinical research is evolving. Now the 20th May is International Clinical Trials Day. The day celebrates the anniversary of first clinical trial by James Lind in 1747 into the causes of scurvy on board the HMS Salisbury. Now his trial consisted of only 12 men grouped into pairs and given a variety of dietary supplements from cedar to oranges and lemons. The trial was only lasted for 6 days, but within that time, there was a noticeable improvement in the group eating the fruit, providing Lind with the evidence required of the link between citrus fruits and scurvy. And the journey started from there and still continues. Now, historically, the long development times coupled with low success rate translated into a high overall cost and risk for the industry. Digital tools are expected to accelerate clinical timelines. However, challenges accompany new technology implementation and evolving trends require agility and investment in a highly competitive environment. Now, what's the current impact and future direction for these new approaches to the therapeutic development? Let's review that very quickly. Moving on, next slide. Now, ICS GCP, which is International Conference on Harmonization and Good Clinical Practice, came into existence in 1996. However, there were a number of medical events and other legislative actions that led to the guidelines we now refer as ICH GCP. Now, in 1906, the first action taken by government to control the way drugs were put on the market is the Pure Food and Drug Act 1906 in USA. Now, it is considered to be the result of very dangerous and even deadly drugs that could have been sold legally just like any other medical goods. For example, such drugs were Grandma's Secret and Cop's Baby's Friend, which contained a huge amount of morphine. Dr. King's consumption cure and Dr. Bull's cough syrup are some examples of drugs which had large doses of morphine and chloroform as well. In this law, it is stated like following: an act for preventing the manufacture, sale, or transportation of adulterated or misbranded or poisonous or deleterious foods, drugs, medicines, and liquors, and for regulating traffic therein. In short, it imposed the regulations on the labeling of products containing alcohol, opium, morphine, cocaine, and some other substances. On each product, it's needed to be indicated which substances were contained in the product and the quality and quantity as well. Now, in moving forward, in 1937, a disaster occurred that proved that this act was insufficient. The so-called sulfanilamide incident in the US. Sulfanilamide was product that used in the battle against streptococci. It was sold as a child syrup by dissolving it in ethylene glycol, also known as antifreeze. Yeah, hence it was a disaster. As a response to this, the US government revised the Pure Food and Drug Act 1906. This became the Food, Drug and Cosmetic Act of 1938. It introduced, besides the stricter regulations on the labeling, safety standards, and required that new drugs were tested on safety before marketing. Now, in 1939 to 1947, until 1947, following World War II and during the Nuremberg trial, it became clear what atrocities the Nazi doctors committed in the name of medical science. During their trial, the Nazi doctors argued in their defense that there were no international law or informal statement that differentiated between legal and illegal experiences on humans. On 17th April 1947, two US doctors who worked for the prosecution of the trials submitted a text with 6 points defining the legitimate research. These 6 points are listed below, then became 10 points and now known as the Nuremberg Code. Although it never had any well-established legal force, it is still one of the foundation stones of ICH GCP. Now, these are the 10 points of Nuremberg Code. First is like voluntary consent of subject. Next is results should be fruitful and not procurable by other methods. Trial design should be based on results of animal experimentation. Next is avoid unnecessary sufferings and injury. The next point is if the researcher believes that death or disabling injury may occur during the trial, the research is not legitimate and should not be done. Next is the degree of risk may not exceed the benefits of experiment. Next is every possible action must be taken to protect the subject from injury, disability, or death. The experiment should be conducted by scientifically qualified personnel. The subject may terminate the trial at any stage. And the last one is the investigator must be prepared to terminate the trial at any stage. Now moving forward, in 1960, thalidomide was used in Europe as a treatment for insomnia, mostly in pregnant women. When the company who manufactured the drug made a submission to FDA to market the drug on the American market. Francis Kelsey, who was an FDA employee, reviewed the application thoroughly and kept it off the market when she discovered that it caused the birth defects when used on pregnant women. Now let's see what happened in 1964. The 10 items in Nuremberg Code were later incorporated in the Declaration of Helsinki, written in 1964 by World Medical Association, an international organization representing physicians. The declaration was a guideline that provided all the documents formed the basis of good clinical practice. But what about harmonization? The onset for this was thalidomide incident. Then in 1970, the consequences was a tightening and amending of the Food, Drug and Cosmetic Act 1938. Eventually, from the mid-1970s, the FDA found it necessary to reject clinical research from other countries since they didn't have the same ethical and safety standards as the US, Europe, and Japan. Each developed their own set of GCP guidelines. Then from mid of 1980s onwards, but the differences between the US, Europe, and Japan remained and did not contribute to the economical efficacy of clinical trials. Pharmaceutical companies had to invest in 3 different trials to globally market one single drug. Then in 1980s, the European Union began harmonizing regulatory requirements. 1989, Europe, Japan, and US began creating the plans for harmonization. Then in 1990, the International Conference on Harmonization of Technical Requirements for the Registration of Pharmaceuticals for Human Use, which is ICH, was created in April 1990 at the meeting in Brussels. ICH had the initial objective of coordinating the regulatory activities of European, Japanese, and United States regulatory bodies in consulting with the Pharmaceutical Trade Association from these regions. And the motto was to discuss and agree the scientific aspects arising from the product registration. Since the new millennium, ICH's attention has been directed towards the extending benefits of harmonization beyond the funding of ICH regions. Then in 1996, an international conference on harmonization was called into and consisted of authorities of Europe, US, and Japan experts, and some other experts from pharmaceutical industries. This led to an international uniform standard to accept the results of clinical trials, as long as they are conducted according to ICH GCP guidelines on safety, quality, efficacy, and multidisciplinary. Now let's move on next slide. So now this slide demonstrates how the clinical research is moving from traditional Western countries such as US, Europe, UK to the East, that is Asia, where more than 35% of world population is residing. Traditionally, 95% of clinical research has been conducted in Western countries and only 5% was contributed by Asia. This is changing, and now more than 50% of clinical research is being shifted to Asia, in more particular countries like India. What does this mean? It will bring more and more jobs in clinical research, and this is something pharmacy professionals should be looking at, an exciting opportunity to make the career in. Moving on, next slide. Now, just to connect the current situation, I'm just showing this slide to show to the major clinical trials conducted on COVID by different pharma companies. They include treatment studies as well as vaccine studies. Now you can see most promising vaccines candidates are from AstraZeneca, which is in collaboration with Oxford University, UK, and another one is Janssen. The clinical trials have begun and we will know the outcomes, I think, in next few months. Now moving on next slide. Now we'll see, let's have a review of how the clinical research is evolving. By now, we know the good clinical practice, which is referred as GCP, is an international, ethical, unified and scientific quality standard with an objective to facilitate the mutual acceptance of clinical data by regulatory authorities in the multiple jurisdictions. The GCP guideline was developed with consideration of the current good clinical practices of European Union, Japan, and the United States, as well as those from Australia, Canada, and the Nordic countries, and World Health Organization as well. The expectation is to follow these guidelines when generating the clinical trial data that are intended to be submitted to regulatory authorities. Broader ICH guidelines are applied by a growing number of regulatory authorities and now includes 16 members and 32 observers. Using the big data available within the public domain related to various regulatory rules, laws, guidelines, and several non-compliances, for example, inspection findings, GCP Compass has studied all this data using the artificial intelligence, deep learning techniques, and GCP domain expertise into a powerful and very interactive online learning tool. This tool has more than 1,000 questions, and each question is connected with the GCP specific area. References are in the form of flashcards and associated non-compliances, wherever it is available. This rich knowledge and powerful use of technology will help user to connect the theory with practical application and then also understand the consequences of non-compliances, which means not following the good clinical practice. Now, with this innovation way, then acquiring such skills through a traditional book or a reading or class type environment of learning, users will learn how to apply the theoretical knowledge into a practice and address some gray areas effectively. Now, this process will user not just prepare to pass the exam, but enable them to retain the knowledge more longer and make learning fun and an enriching experience. Now moving on next slide. Now we'll see what are the benefits. So the global clinical trial market size was valued at $46.8 billion US in 2019, and it is still expected to grow at a compound rate of 5.1 from 2020 to 2027. Globalization of clinical trials, technology evolution, and demand for the contract research organizations to conduct the clinical trials are further projected to drive this growth. Most CROs are struggling with CRA retention or ongoing challenge and not a new to this industry, has experienced turnover at about 20% for the last 20 years. The cost of onboarding CRAs are also increasing, and with the advances in technology within the clinical research, like risk-based monitoring or virtual trials, it is even more important for CRAs to gain a deeper, better, and quicker understanding of new world of monitoring alongside of changing regulatory landscape. This has not only impact on cost associated with the onboarding, but Also with the time required to onboard the CRA and cost associated with poor delivery of monitoring. Like by using, we expect this in turn will help in about 15 to 50% reduction in CRA onboarding cost, billable time, and cost of poor quality. Moving on next slide. Now let's understand what are the unique features the GCP Compass, which is virtual learning environment platform, can give. GCP Compass covers the broader area of GCP, not just limited to ICH GCP, but it also covers other guidelines such as US FDA guidelines, MHRA, which is Medicine and Healthcare Products Regulatory Agency in UK, EMA, which is European Medicines Agency, PMDA, which is Pharmaceuticals and Medical Devices Agency in Japan. GCP Compass provides most up-to-date information on GCP areas, and all questions are built around not just based on historical information, but also on the latest information. And questions are also multiple choice type. GCP Compass has used a concept of bite-sized flashcards, which basically uses information taken from one of the 13 key GCP databases. These flashcards are prepared in such a way so that the user can learn topics byte by byte without too much information overloaded at one time. GCP Compass has proactive way to learn GCP in a fun and interactive way without taking burden of classroom or learning exam style environment. When the user provides the right answer, tool encourages user to inform that it's a right answer if it was not a right answer. Tool also provide the information what is the right answer. More importantly, tool will give them inform the user why it is the right answer by giving immediate reference to the specific section of GCP for enhanced learning. In addition to that, tool has also connected any inspections findings associated with that particular area so that user will know the consequences of not following GCP in terms of data integrity and patient safety. Now, the tool has inbuilt algorithm by which when the user answers any questions incorrectly, only these questions are repeated with the answers randomized. That means the sequence of answers will not be the same when user attempt this question next time. This will continue until the user gives the correct answer for all questions assigned for him or her in practice test. The users will have ability to see their own progress through an interactive talent profile, built and designed with the advanced analytical techniques, and then personalized for each individual user. More importantly, it will not be static, and therefore, as you attempt more questions, it will show your real-time progress and also direct which areas you have done well and which areas you will need to focus on before attempting the actual test. Now, the GCP Compass also have introduced a GCP test to test user knowledge using two types of tests. One is like traditional time test, but the other test is also an open book policy test, where the user assessment is then done with the data analytics techniques by using speed and accuracy of their response for each of the question. Once the user successfully passed the test, they will be honored with e-certificate, which you can see here in the picture. As a part of ongoing enhancement of tool, we will introduce additional questions of different types and more relevant to the current scenario. There is a plan to make an Android and iOS based app version for the tool in near future. It is also important to note that the questionnaire in the tool will not be static, and we will ensure it's up to date with the changing regulatory landscape, and therefore you will always have something to learn and learn the right things every time. And that's our commitment from GCP Compass. Now you can see in the screen, like this tool also help you to see your own progress and identify the area that you have done well, and also where you need to improve your skills in the clinical research. Now we'll see how it works in a short demo now. Let me switch on for the another computer. Give me a few seconds, please, while I'm switching off the another screen.
Yeah, sure, sure.
Thank you. Can you see my screen?
Yeah, we are able to see it.
Yeah, thank you. So this is the home page. You can see. As you can see, we have received the accreditation from the Council for Clinical Research Education. User can have a free test where you can see here. Well, for that, we need your details, and it will not take more than 20 seconds to fill, and then you are able to give the free test. Now let me show you the actual test. Let me log in. So here you can see, as I explained previously, you can see the dashboard, and these are the particular areas of GCP. And once you keep on answering the question, this bar will change from red to amber and to green. So when you are doing the practice test, it will keep on showing you which areas you are progressing, and this will throw you the questions until all the bars will turn 100% green, and then we'll see how the practice test is. So this is how the test looks like. So you can choose any option and you can confirm. So system will say this is wrong answer. So let's see what's the correct answer for this, which is in flashcards. So this will show this is the answer, and in this particular area of ICH GCP, you can see this. Now let's try next question. Again, this is wrong answer, so you can have a flashcard. Let's try next question. Let me try one more question. Yeah, so now you can see when you give the correct answer, the system will show. So this is the correct answer. And you can see, as I showed you previously, in the flashcard, you can see. So if it was your guess, still you can see what is the correct answer and which area is that. And now you can see here, we have inspection finding as well. So for not following the GCP, what happens? User can see this inspection finding as well. So now, once you do this practice test and you got the 100% efficiency, the system will ask you to go to the actual GCP test. So as I said, we have two kind of tests. One is time-based and the other one is open book policy. So when you give the test and still you think you need more detailed knowledge, we have the online training available for this. And as you can see, we have covered this ICH GCP area, then the new amendments in ICH GCP, which is E6R2, then 21 CFR Part 11 and 21 CFR Part 54. These are US regulations. Then MHRA, which is UK regulations. And additionally, even though it is not part of clinical trial, still now GDPR, the data protection is coming up, and that's why we have added this point as well. Now you can see if we have done this ICH in subsections. So if you want, you can click on any section and you can directly go to that particular section if you are looking for. You can move forward and backward section if you like. Then in that one point, you can see subsections. We have subcards as well to make it easy. The another feature is in search word, like you can type whichever you are looking for. Like if you are looking for any ethics committee or related to that things, then this all sections will come with that result. If you are looking for, suppose, informed consent and you search, then it will show all the sections which it's containing. Now I would like to show that we have useful links here provided in our tool, and whatever the information mostly I have presented in the slides, all this is available in that useful links. So if anybody would like to check, they can have this in the system. Now I would like to summarize a small video which I will check if it is working here directly to YouTube. Let me have a look. So you can have a look. This is a short demo video. I will start.
Switch.
Now bear with me. Let me switch the screen again. Now you can see in the initial phase, GCP Compass is offering 3 types of membership. So as a guest or visitor, you will have the free access to useful references, material, and latest update. We are coming with some GCP news channel as well and some short introductory videos. Once you are convinced with yourself that this is a right tool for you to learn GCP, you will be able to request for actual demo of the tool and practice some questions by creating your profile. Now, after you have tried out the sample questions, our tool will analyze your data using the deep learning techniques and then will suggest you the best learning level, which is either basic or professional, to start with us as a part of next learning journey. Now you can then subscribe to one of the membership to learn the GCP in fun way to test your knowledge and gain a better insight with the latest information. Now moving on next slide, we'll see what are the different roles available in a clinical research. Now, as you can see, this slide shows the employees from the top clinical organizations. The top CROs, you will be surprised to know that these organizations had almost no clinical research staff in India until 1990. And now they employ between 20 to 30% of the global task force from India. Excuse me. As India has the talent and we have our education courses in English, which is the advantage to us as compared to the other big population countries like China. Moving to the next slide. Now, this slide shows you the quick snapshot of various clinical research roles. Like as you can see, you can either enter in clinical operations if you like to be a person in operations. You can start with the project assistant or project associate and then move on to the next level. If you like management, you can go to the project management, or if you like the regulatory area, you can enter in regulatory affairs. Someone can go in medical services, or if you are business minded, you can go in business development. If you like the hospital setup, you can start as a clinical research coordinator. Then somebody can go in drug safety if they are interested in drug development, or if anybody is more handful with data, they can go to the data management. And if you are interested, there are some opportunities in labs as well. Or quality assurance is the another side where you can do auditing. Now, thank you so much and hope you enjoyed the presentation and discussion. With this, I would like to end my talk and thank you once again.
Thank you, Dr. Patil, for a very nice presentation. Actually, we found the data that you have compiled and shared with us was really very informative. The way you have discussed the drug development process, clinical research, opportunities in clinical research, particularly for pharmacy profession, it's really very, very useful for all our participants and research scholars, other students. Thank you very much. And I would request the participants also, they can ask the questions.
Yeah, sure.
Yeah.
I'll try to, yeah.
Yes. Participants can unmute yourself and you can directly ask your questions.
Yeah, I'll try to answer the question. If not, my expertise are here. I can ask them to answer.
Yeah, otherwise presentation was very clear, Dr. Patil, and everything was very clear.
Thank you.
Yes, any participants want to ask any question? From participants?
Yeah, even the participants can also send their questions to
Directly to ma'am.
We will share the questions with the expert also. So if there will be any other questions, if you want to ask now, you can.
If you don't mind, I would like to add one point. Like when the previous speaker, somebody asked them about the curcumin study.
Yes.
Yeah, so other than wound healing, if you are interested, you can try on dementia.
Right.
Okay, ma'am. Okay.
Yeah, so that will be the upcoming one. Like now, the Alzheimer's and dementia is quite progressing, and you can that can be the area of interest where you can try.
I really appreciate your input in this, Dr. Patil, and
Thank you.
I think we would look into this. I hope that this is not already recorded online. Thank you so much.
Yeah, no worries.
I think some studies have been done for curcumin in this area. You can just check it up for cognitive performance.
Mm-hmm.
So probably you can take it further from there.
For anti-inflammatory activity also, it can be used.
Yeah, anti-inflammatory as well as wound healing is already recorded. I think many number of studies has been done on curcumin already.
Yes,
And
yes.
these two are mainly 80 to 85% of these being recorded online. That is why I was asking.
Yeah. Everything starting from pain to cancer.
Yes, sir. Yes.
We will just be looking for the further scope in these areas.
Yes. Dr. Wakude, you want to say something?
Well described and good presentation by Dr. Rajeshri. Thank you very much. And I
Thank
think
you.
it is good one for the students, our research scholars especially, who are in the clinical research area, as well as teachers who are mentoring them. Now in 8th semester, we are having this PCI syllabus. They need to choose one subject, either clinical research, medicinal chemistry, pharmacovigilance, that marketing, regulatory affairs type of thing. So in that case, it is very easy for the students to go through the content of this your website GCP Compass, and they can try themselves, learn themselves, and explore what are the things which are going at the international level in the clinical research field. So that is
Yes.
very good aspect. In future, whenever you come to India, you are most welcome to our university, DIPSAR and DIPSAR. So
Yeah, it would be my pleasure.
because we are having MPharm in clinical research, MPharm in regulatory affairs. So these are the good fields where you can be of great help to all of us. Even our Vice Chancellor, sir, our Professor S.K. Gupta, is doing
Sure.
wonderful work in the clinical research, pharmacovigilance, and pharmacology. So they will be glad to meet your organization, and in future, if possible, some type of interactions we can increase at that level. So I wish all the best. Thank you very much. And the teacher candidates, delegates who are especially from the pharmacology background or want to pursue themselves in the higher studies with some pharmacological or some pharmacovigilance or clinical research type of additional courses. So they can go ahead with this type of courses. Like in Jamia Hamdard also, IHMR also, Jaipur, they are doing, but that is offline courses sometimes, and it becomes difficult for the individual teacher to go and attend that thing. So as a online part also, this your course or modules may be helpful to students also. Thank you very much. Thank you.
Yeah, thank you so much. We are currently working also some of the universities in UK. So we are trying to make it as easy as possible and fun loving activity for the kids so that they can enjoy and acquire the knowledge.
Yeah, again, thank you, Dr. Patil. Anushma, to you.
Thank you, ma'am, for the informative session. Now, I would like to request Dr. Geeta Agrawal to kindly felicitate Patil ma'am and Mrs. Manju Vyas ma'am to kindly share e-certificate.
Thank you, ma'am, for joining us, and please accept this from our side.
Thank you so much.
Yeah, always welcome. And I would like to congratulate all the organizers, Manju ma'am, Bakodi sir, Bodla sir, everyone. So congratulations to you too.
Thank you, Dr. Geeta, for your participation. As I said, it has added the flavor to the session.
Thank you, sir and ma'am, for joining today's session. Now we will be wrapping up for day three session here, and tomorrow from 10 o'clock sharp, session will be starting. Kindly join us. Thank you.
Thank you, Dr. Patil ma'am. Thank you very much.
Thank you.
And thank you very much for Dr. Geeta Agrawal also for chairing the session.
Thank you, ma'am.
Thank you, Dr. Geeta. Thank you, Dr.
Thank you once again, and I will stop here.
Okay, ma'am. Thank you very much.
Thank you.